Right. Uh, so this second part, which is second unit, we have three chapters: morphology, anatomy in flowering plants, and then structural organization in animals. In which we have detailed discussion on cockroach as well as frog. Right. So before going into this chapter, just write this one. Catherine Issa. Sometimes the questions on this introduction part are also being asked. Last year also there were some questions, if you remember. You better write this. Uh, she published a book, Anatomy of Seed Plants. This is the book she published. This one is actually considered as the Webster's of plant morphology, plant biology. Uh, in case you don't know what is Webster's, Webster's is actually a dictionary. Okay, so this book is considered as the Webster's of plant biology. This book, Anatomy of Seed Plants. It was published by, it was actually written by Catherine Issa. So just write this contribution. Uh, there's a discussion about her in the beginning of this chapter. So just look into that later. But important thing is this, just note it down. Right. Next two. What's the meaning of morphology? I mean, etymology of morphology. Uh, okay. It's not just external appearance. Anyhow, let's see this. When we say morph, morphph, morph, it refers to form. morph, morph is form. Logos, as you know, study. So basically, it is study of form. Right. There are two branches in morphology. One is internal morphology. The other one is external morphology. Right. Now, basically, when we say morphology, by default, we are actually talking about external morphology only. Right. But when you see internal morphology, we call it normally anatomy. So basically, in morphology and anatomy chapters, we are studying about the form. Right. And again, what is meant by this form is form refers to structure, shape, right, color, right. All these features together is what we call the form. So whether you are studying the internal form or external form, we have two different branches. So in this chapter, which is morphology, by default, we are actually studying about external morphology. Whereas the internal morphology is what we call anatomy, which is the next chapter is. So just write morphology is what the first part is. Right. So in this, we'll be studying about root, stem, leaf, uh, in the part which is vegetative morphology, right? And then we'll discuss about inflorescence and flower in the next part, which is reproductive morphology, right? And once we finish discussing this vegetative morphology and reproductive morphology, we have discussion on some plant families. So totally, this chapter is having three parts: vegetative morphology, reproductive morphology, and plant families. See, the thing is, whatever we study in vegetative morphology and reproductive morphology, that will be repeated in this. So we will not study anything, any new term in the plant families. Okay, only thing is, okay, for example, some family like Solanaceae is there. In this Solanaceae, what are the vegetative characters? What are the reproductive characters? And of course, examples. So only thing that we will be studying in this part is examples. Otherwise, every term that we have studied in vegetative morphology and reproductive morphology will be repeated here. Okay, that's why practice well this part, vegetative and reproductive part. Then plant families will be easier one. So we'll start with the first one, which is vegetative morphology. Right. Again, before jumping into this root, stem, leaf, and all, let us understand one important part inside the plant, that is embryo. There are some technical terms related to this. Uh, we need to understand that, then we'll proceed to that different parts in the plant. So when you see the embryo of dicot or monocot, understand this thing properly. There are some terms which you have to learn here. Now say this is the embryo is this axis, the axis which I have shown here. This is embryonal axis. Now basically the one which I have shown is embryo. This axis is embryonal axis. Embryo is associated with cotyledons. Embryo is associated with the cotyledons. See, whether one cotyledon is there, two cotyledon is there, is what the difference is between dicot and monocot. But basically, two cotyledons will initially develop. Only thing is, in the case of monocot, what happens? Only one cotyledon develops well, the other one will be reduced. So that's why let us not worry about dicot and monocot now. So it is like two cotyledons are present associated with it. How much developed we'll see later. So these are the cotyledons. Right. Now hope you are clear. The one which you see is the embryonal axis. The white colored one which I have shown is the embryonal axis. This one is embryonal axis. Right. That axis is what I have shown here. This complete axis is embryonal axis. It is associated with the cotyledons. Right. See where it is associated with the cotyledons. It is associated with the cotyledons at this level. I'm writing a red line here. This is the level at which embryo is associated with the cotyledons. Right. The complete part above this one is what we call, yes, anyone? Epicotyl. See the name EPI above cotyle is cotyledon. So basically it is above cotyledon. So you're trying to say that this is the part of the embryonal axis which is above the cotyledon's level. Exactly the reverse is this. This one is hypocotyle. Right. Now in this, the terminal portion of this epicotyle, exactly this part. Yes. This part is what we call plumule, whereas this part is what we call what? Hmm. Right. Copy it if you are clear. There is a lot of discussion on this throughout this chapter, so be clear with this terminology. Epicotyle is which part, hypocotyle is which part, plumule radical. Finished? Okay. Then types of before that, let's write root system. The first one is root system. Right. I mean, this is the first one type. Types of root system. Okay. Just try to understand. I'll just give the flow chart here. There are three different types of root systems. Taproot system, fibrous root system, and then adventitious root system. RS is root system, I'm just writing in short. So there are three different types of root system: taproot system, fibrous root system, adventitious root system. So the difference among these three is this: in taproot system, what happens is radical. Now this one, will develop into first primary root. Radicle develops into primary root. This primary root will give branching, right? It is branched. We call it lateral roots. So basically, if this is the primary root is, from this again there will be formation of lateral roots. Right, from this there will be branching further. So the main root system which developed from the radicle, that is primary root, will be giving a complete branching network. All these together is what we call taproot system. Right, now these lateral roots are of different orders. This can be secondary roots, tertiary roots, etc. I hope you understood. The first one is primary. Primary will give branches which are secondary. Secondary will be branched to form tertiary. Like this, primary root is giving basically branching network. All these together is what we call taproot system. Right, this you can see in dicots. Right, there are many examples. One example given here in this part is mustard. But otherwise, all dicots are actually examples for this. Now, see the difference with this fibrous root system. Here, in fibrous root system, the radicle develops into primary root. Now, everywhere, radicle only will develop into primary root. Right, but this primary root is short-lived. It is short-lived. And later what happens in the place of this thing, short-lived means what? It will disappear. Right, this one will disappear. After that, in that place, there will be formation of many roots. So it's basically like this. At the base of the stem, whatever the primary root, if this is the primary root is, this primary root will disappear. In the place of it, there will be formation of many roots like this. That's why it appears like the fibrous network. That's why the name fibrous root system. This you can see in the monocots. Right, again, monocots means all monocots will come under this. Right, here in this such part, example that is given is wheat, or it can be rice, any kind of monocots. Next, adventitious. Now see what is common thing between this taproot and fibrous root system is radicle is the one which is giving the primary root. But only difference is what here? Here the primary root is persistent, will be giving the branching network. In the second case, primary root actually disappeared. But definitely primary root developed in both the cases from radicle only. Here, adventitious roots means, first of all, adventitious means abnormal. Now, anything which is not normal, we call it adventitious, wherever. The technical term of adventitious means abnormal. So here, roots develop from parts other than radicle. So if the roots are developing from some other part of the plant, not from the radicle, then we call it adventitious root system. Now, for example, the best example, banyan, if you see, banyan plant. There you can see the tree is having very large branches. From these branches, you can see roots developed. Yes, what do you call these roots? Anyone? These roots are what we call prop roots. These prop roots are the examples for adventitious roots. Right. The same way you can see in the monstera, in the monstera plants, and also in the grasses. Right, you can see roots developing from some other parts, not from the radicle. Those roots are what we call adventitious roots. So these are the three different types of root systems. If clear, copy it neatly. If not, just let me know. Just write the flow chart. Right. Next one. Tarun, finish copying.
Yes, sir. Yes, sir.
Next one. Regions in root. And again, we'll write everything in single figure only. So just see that. Right. The first one, in the tip of the roots, as we know, there is meristem. The meristematic cells are present in this. This is what the meristematic zone or meristematic region. This is the first region. Right. As the plant is this root is growing inside the soil, to protect this meristematic region, there is a root cap. The function of which is to protect this meristematic region. Right. As you know, this meristematic region is the one where the cells are actively dividing. So when the cells are actively dividing, they will be small. Here the cells are small. Right. Primary cell wall will be present. Right. Primary cell wall is present means what? Only the cell wall is very thin. There will be thin cell walls, and any actively dividing cells, cytoplasm will be dense. There will be dense cytoplasm. So basically, this is the description of meristematic cells only. And therefore, the cells present here will be small with a thin cell wall and dense cytoplasm. They are basically actively dividing, rapidly dividing. Next, here one logic you need to understand. See, if this is the meristematic zone, listen properly, there is a concept here. In the meristematic zone, as we know, new cells are added in this part. Right? But when these cells are undergoing differentiation, some cells of it will be undergoing differentiation also. Any new cells formed will undergo differentiation to form permanent tissues. Now, the point is here, towards the proximal end of this meristematic zone, that is towards this side, they will be undergoing maturation or differentiation. Means only the cells which are present or added towards this will be undergoing maturation, not the cells present inside this. The cells which are present inside this will be having active cell division only. Only the cells present towards this side will undergo maturation. So what you need to understand here, continuously new cells are added, right? But the cells which are present towards this side will be undergoing maturation. Means what? This region will remain constant. If not clear, just ask now itself. This zone, the meristematic zone size, whatever is there, this size will always remain constant. Why? Because new cells are added, but the new cells added towards this side are undergoing maturation. So they are not the part of meristematic zone now. So always meristematic zone size is constant. Now how, even though new cells are added, still the size remains constant means, hope you are clear. Right. So this part is what we call region of elongation. Elongation region. Now, why elongation region? Because this meristematic region is remaining constant. It is not responsible for the growth of the roots. The size of this always remains constant. New cells are added into this only, right? Where they are undergoing maturation. So the length of root will be only in this part. Right? That's why this is called elongation zone. Right? Can anyone tell the technique which is used to understand this differential growth? How do you know that growth is present only in this region, not in the other parts? What is the technique that we have discussed about? What line? Hmm. Very good. In the parallel line technique, which we have discussed in plant growth and development. By using this, we have understood that every part of the root will not be having the equal growth. Okay, growth is present only in this zone, that's why it is elongation region. Right? So what is happening in this region is cells are undergoing differentiation. Undergoing differentiation, maturation. So during maturation only vacuolation will occur, right? So here vacuolation will occur. Vacuolation will start. Okay. And finally, this is the part where the cells are in the highly matured state, which is what we call maturation region. Okay, and what is the specialty of this part is this is where you will see the root hairs. The root hairs are present only in the maturation region, not anywhere else. Keep in mind. Right. So this is about regions of root. Right. The next one, stem. See, actually, there are modifications in the root which used to be there earlier. If required, we'll discuss later. Otherwise, you just prepare these things first. So let us see the syllabus. If mentioned, we'll see that. Otherwise, means once the bulletin is released. Otherwise, you can just prepare only this. Right. Now coming to this stem. Now there is no big discussion. These very small things are there. First of all, stem will develop from which part of the embryonal axis? Epicotyl in the plumule. Right. Plumule of epicotyl. Right. Next, this is what do you say? Ascending part of plant axis. Right now, as you know, in the plant axis, this is the one which is growing upwards. Therefore, it is the ascending part of the plant axis. Whereas the root is, though we haven't written here, root is what? It is a descending part of the plant axis, whereas this one is ascending part is. Right, next. Positively, positively what? Hmm. Positively phototrope, PIC or PHIC? Is it PIC or PHIC? Hmm. PIC, not PHIC. PHIC means nutrition. Okay, PIC is growth, right? So it is growing towards the light. That's why it is positively phototropic or negatively geotropic. negatively geotropic. And how do you differentiate stem and root? Okay, good. That's surprising. Mm-hmm. From which develop from birth only anyhow. Hmm. Is the potato is stem or root? Is the potato stem or root? Hey, what happened? Sure. Tarun, is it clear for you? Clear now? Now, potato is modified stem. But how do we know that it is modified stem? See, first of all, it is not green in color. It is brown in color, right? Root. Right. It is not having buds. Will it be there or not? We'll see. Next, it is not aerial. It is underground. But still, how do you know that potato is stem? Okay, this is what the feature is. Stem consists of nodes and internodes. And nodes. So this is the unique feature which you don't find with the roots. Right. Now, hope you are clear. Nodes and internodes is what the difference is. So, what these nodes and internodes are in the stem? Right. Now, these regions are what we call nodes. From these nodes only, branches, branches or the leaves will develop from only the nodes. And the region between two nodes is what we call internode. This one is internode. Inter is between nodes. So between two nodes, that region is what we call the internode. So presence of nodes and internodes is what the characteristic feature of only stem, which you don't find in the case of roots. Right. Next, the buds which develop here, these buds can be terminal buds or axillary buds. They can be terminal buds or axillary buds. Right. Apart from this, you know the functions of this. Um, it will be helping in the photosynthesis. Will be helping in the transport part. Right. It is protecting different parts. So all these things you can just go through it quickly. So the functions of root as well as stem. Right. So when it comes to these birds, terminal buds are produced in the terminal portion of the branch. Right. Whereas the branches, this is the branches. In the axilla of these branches, if bud is produced, this is axillary. So the buds can be terminal or axillary. Hmm. Next one, leaf. Right. Now you know the structure. Mhm. Right. What do you call the stalk of the leaf? What happened? What do you call the stalk of the leaf? No, no, no. Stalk.
Petiole.
Petiole. Petiole. This one is the leaf basis. This is leaf base. The stalk is what we call petiole. Right. And this expanded part is what we call lamina. Uh, all this expanded part is what we call lamina. Right. Next, inside the leaf, there are veins. Right. The main vein is what we call midvein, which is branched to form veins. Veins are divided to form veinlets. So this main midvein will divide into veins and then into veinlets. So basically, all this is the branching network of veins. So what is the specialty of these veins is vascular tissue is present in this only. Xylem and phloem, complete vascular tissue is present inside the veins and veinlets only. Right. Next, in the axil of the leaf, in the axil of the leaf, there can be axillary bud. There can be axillary bud. Right. This axillary bud either develops into branch, that is vegetative branch, right, or it can develop into a flower. Now, see, as we know, flower is the modified branch only. Sorry. As the flower is a modified branch, either branch will develop in the vegetative growth or there may be flower development that will develop from this axillary bud only. Right. Next. Here at the base of the leaf, there may be leaf-like structures. It's not the universal feature. There may be presence of these leaf-like structures. Where at the base, like this, one, two. There will be two leaf-like structures. These are called, what do you call this? Hmm. Stipules. How many? There will be two stipules developed at the base of the leaf. Now see, basically, if stipules are present, we call it stipulate leaf. Stipulate leaf. But there are ex-stipulate leaves also. Right. If it is absent, we call it ex-stipulate leaf. Right. And not everywhere. In the leguminous plants alone, you will see this thing. The leaf base is actually bulged, swollen. Right. Swollen, if swollen, we call it pulvinous. Pulvinous leaf base. Only thing is the leaf base which is present here, it is swollen. If it is swollen, it is called technically pulvinous leaf base. This we are going to see in the Fabaceae family. That is Leguminosae family. Right? So this is about different parts of the leaf. Next, this arrangement of veins is what we call venation. Now, venation is arrangement of veins. There are two possible arrangements of veins you can see. One is, see the arrangement will be like this. There will be a main vein which is divided. Again, these are divided further. Right. Like this, there will be branching network of veins and veinlets. This is what we call reticulate venation. Right. Now see, wherever the term reticulate comes, what is the meaning of reticulate? Very good. Network. In the endoplasmic reticulum also we have seen the same term. Reticulate means network. So what is this reticulate venation is? Veins are divided, redivided to form a network. Therefore, it is called reticulate venation. This is what you will see in the dicot plants. But in the other venation called parallel venation, there will be many veins which will be like this inside the leaf, and there will not be much branching of it. So it's basically parallel, parallel venation. So why parallel venation? Because here veins running parallel to each other. That's why the name parallel venation. So you don't see much branching network and all in this. This you can see in monocots. Right. The next one, phyllotaxy. And again here, taxi is from taxis. Taxis means arrangement. Now even the term taxonomy. What the meaning of taxonomy is? Taxis is arrangement, nomos means rules. Taxis plus nomos. Nomos means rules. So basically arrangement rules is what taxonomy or classification, right? The same way here, phyllotaxy means taxis is arrangement. Phyllo means leaf. So it is basically arrangement of leaves on the branches or the plants. So how these leaves are arranged, arrangement pattern you can say. So there are three different possibilities here. The first one, see these are the nodes. The arrangement will be like this in the first one. If one leaf is here, there is only one leaf at each node. One leaf is here, the other one is here. Next here, next here. So leaves are arranged alternatively. This is what we call alternate phyllotaxy. So what is the unique feature of this thing is one node will have only one leaf. Right. Next step. Here there are two leaves. So leaves are present opposite to each other. Therefore, we call this one opposite phyllotaxy. The kind of phyllotaxy is opposite. Here, one node will possess two leaves opposite to each other. Right. And coming to the last one, here there are many leaves at each node. Like this, there are many leaves at each node. So these many leaves will be forming a whorl. A whorl means it's a kind of sheath. That's why this is called old phyllotaxy. old phyllotaxy. So what is it? Here one node will possess multiple leaves. These are the three different types of arrangement of leaves. Right. Apart from these examples, only NCRT examples are enough. Don't go beyond NCRT. Okay. China rose, mustard, and sunflower. Opposite, very important example, calotropis. Calotropis plant. Apart from this, if you have guava plant in your garden, you check it. Guava. In guava also you can see the opposite phyllotaxy. Next, old phyllotaxy you can see in nerium. It's not given in the textbook. So Alstonia. Alstonia is one example given. If you are clear with the logic, no, the names are very easy. Right. The next one, types of leaves. See, here also you need to understand the concept properly. And listen properly first. See, I'll take three different possibilities here. Right. See here, the margin of the lamina. This is the lamina, right? Lamina's margin is there, no? It is entire. If you don't understand this thing, will be difficult later. Just see, entire means there is no incisions. Leaf is not having any kind of incision. A smooth surface means it is complete surface. That's what we call the entire. The second possibility is here the lamina is having incisions. Right here, there are incisions. So lamina is not entire, it is having incisions. But in both of these two cases, either incisions are absent entire, or even if incisions are present, they are not reaching the midrib. This one is the midrib, right? Midrib means main vein. The main vein is what we call midrib. So in both the cases, midrib is intact, and these incisions, even if present, they are not reaching the midrib. But if you take the third possibility, this one is a midrib base. Incisions have reached the midrib. They have divided this lamina into small leaves now. Right. Now keep in mind all this is one leaf. Right. In this, these structures are what we call leaflets. Leaflet means small leaf. So what is the difference among these three? In the first two cases, which are called simple leaves. This is simple leaves. So what is simple leaf? No incisions, or if Incisions are present. They did not reach what midrib. Then that leaf is what we call the simple leaf. But in the next type of leaf called the compound leaf, incisions have reached the midrib. Incisions reached midrib. Right? Dividing what? Lamina into smaller parts. What do you call the smaller parts? Leaflets. Is this concept clear, everyone? Right. So draw it and write this explanation. Simple leaves and compound leaves. So in simple leaf, the lamina will be either entire or incised. But the incisions did not reach the midrib. But in the compound leaf, incisions reach the midrib. Finished? Okay, finish it. Meanwhile, just think about it. If you see some leaf like this on the plant, see this one on the plant, how do you know that these are leaflets? They are not leaves. The same structure can be seen in opposite phyllotaxy also. So when they are present on the plant, how do we know that they are leaflets, not leaves? Is my question clear? Think about it. Okay. See this here. Hmm. Is Kanishka, you answering? Mm-hmm. But how do you know that it is not branch or stem? See, when you see this thing directly on the plant, how do you know that it is not branch? It is leaf. My yes or no, that can be branch also, right? Who knows? So the difference is this. See what we have seen here in the axil of the leaf, there is bud. What is that? Axillary bud. Okay. So where do you find the axillary bud in this? Understood? So if this is how the leaf is, axillary bud will be present here, right? Yes or no? This is where the axillary bud will be. Right. Will the leaflets have the axillary buds? That's the difference. Right. Next one. Uh, Now compound leaves. These are of two different types. Right. See the difference in the first one. The one which we have drawn is the same only. Right. And this is how the leaf is. The midrib. This one is a midrib, right? So there is one common axis to which all the leaflets are attached. So all these are the leaflets attached to one common axis. A common axis is what? Midrib, right? This one is midrib. Now this midrib is what we call rachis. Now don't confuse, rachis means it is midrib only, but in the compound leaf, when it is bearing leaflets, we call it rachis. Right. Now see the other possibility. Uh, this one is the petioles. This part is the petioles. Now in the second possibility, see properly, this is the petiole. Like this, many leaflets attached to the terminal part of the petiole. There is no rachis. There is no common axis. Why? Because all leaflets attached to the terminal portion of the petiole like this. This is what we call palmately compound. It's like palm like. That's why palmately compound. The first one is what we call pinnately compound. Pinnately compound. These are the two different. So in a palmately compound, no ratches. There is no common axis. All leaflets are attached to a common point that is here. So no ratches here. Right. You can observe neem plant. Neem will be having this structure only if you recollect. That is actually pinnately compound one. Example for this palmately compound is silk cotton. Right. My finished. Will close the session here. But tomorrow you come practice these flow charts and figures, whichever I have given in the notes. Only flow charts and figures, whichever the figures I have given, the diagrammatic representations. These. Everything is not there in the book, but you practice this thing. Okay. So tomorrow you'll have the test and we'll move to the next part that is reproductive morphology. Right. Tomorrow we'll see this thing. Yes, yes, I'm opening. Yes. Okay. Okay. Here the question is about what? All of the given characters have no exceptions except. So if you take the feathers in birds, every bird will be having feathers. There are no exceptions for this. Flightless birds are there, but featherless birds are not there. Mammary glands in mammals. Every mammal will be having mammary glands. But if it is a functional mammary glands, then this also can be the answer. Keep in mind. Okay, if this is the case, it is also exception. But otherwise, it is given as just mammary glands. Functional or not does not matter. Next, three-chambered heart in amphibians. There are no exceptions. Right, but heterodont teeth in the jaws of mammals means what? It is not the universal feature, right? Every mammal may not be having the same. Most of the mammals have, right? But there are some mammals where there are no teeth. There are some mammals with some other kind of teeth. Examples not required, but there are exceptions. That is reptile, madam. That is reptile. In reptiles, yes, there are exceptions. Crocodiles, right? Don't be in hurry, Kanishka. Be clear. Be clear. 125. Christies has to swim constantly to avoid swimming. Yes or no? Right, because it is chondritis, no? Absence of air bladder leads to the loss of buoyancy in the animal. See, when there is air bladder, they need not to, I means they need not to swim constantly because it will give the buoyancy, no? Because the air bladder is absent, they have to swim constantly. Therefore, it is a correct explanation. That's what absence of air bladder leads to loss of balance in the animal, no, in this animal. Animal means which animal you are assuming, ma? Why? Because it is a continuation of this only, right? Now don't take the things in that way. Be clear. So here animal means we are talking about one animal which is given earlier, right? That is this only. And that too it is given as the animal, not animals. Yes, next one. 139. What's the doubt? Here, check the habitat of Pleurobrachia, Cucumeria, and Exocetus. Pleurobrachia is so marine. All animals are marine. Cucumeria, marine. Spongilla, is it?